Welcome back. It's Intro to Physical Anthropology. I'm your instructor, David Leitner. Today, we're going to continue our discussion of DNA function with its second major function. First is replication. Second is protein synthesis. That is, creating proteins. Proteins, as we'll find out, are if cells are the building blocks of organisms, of life, then proteins are the building blocks of cells and everything that goes on in them. So, without further ado, let's go ahead and get started. I should say, actually, too, you'll note in the on the course website, there's also links to a couple of really helpful animations to describe transcription, translation, and also something else we'll talk about, which is alternative splicing. So be sure to look for those. They're really short videos, but if you're more of a visual thinker, they give you a better idea. All right, so here we go. So proteins. Proteins are a kind of very complex molecule. They're usually chains of shorter molecules called amino acids. These molecules are involved in everything from, every tissue in your body is made of proteins, right? Every chemical reaction has something to do with proteins in your body for the most part. Enzymes, which are special proteins that metabolize, that control control how chemical reactions occur, are generally made of proteins in the body. Hormones, all of these things. I can go on and on. In fact, actually, the current sort of big push in molecular biology, after in the 1990s we decoded the human genome, now we're talking about decoding the human proteome. That is the entire combination of proteins that our DNA can produce, can code for. And that is a much larger sort of task, that, as we'll find out. Okay, so proteins have very complex structures. As I said, they mostly come in chains, like you see at the top here, but then they can either sort of fold up or coil up, and they form these wiggly wobbly three-dimensional shapes that both have chemical and physical properties that allow them to do the things they do. Now, protein synthesis occurs out here on the endoplasmic reticulum, out in the cytoplasm, at the sites of these little red ribosomes that I've colored in. Okay. What the ribosomes are going to do is they're going to pull in amino acids and hatch them together, one after the other, into a chain. That chain is a polypeptide. That is, it's made up of many amino acids in a long chain. The chemical properties of the resulting protein are going to be determined partly by the exact sequence, well, mostly by the exact sequence of the amino acids, but also the way that those amino acids cause the protein to fold and twist as well. There are over 20 to the 200th combinations of possible proteins for a typical protein. That's assuming we've got 20 different amino acids, and the average protein is somewhere around 200 amino acids long, though it can vary greatly by, factors of 10 or more. So, but as a rough estimate, this seems to be, it just gives you an indication. This is a big, big number. There's just another image of the chain. Each of these little, links in the chain. Hey, come back here. There we go. Each of these little links in the chain is a different amino acid, as you can see. Now, as I said, there are 20 different amino acids, plus there's a class of amino acids. There's each of those amino acids is coded for by 3-letter sequences in the DNA, plus there's a class of 3-letter sequences called termination codons that simply tell the process to stop. That'll make more sense in a minute. So, if you've got 4 possible nucleotides and 3 letters at a time, then these 3-letter combinations, which we call codons, that's another name for it, these 3 base sequences, you've got a total of 4 to the 3rd power, that is 64 combinations of different amino acids of different bases that can be combined in this code. That said, there are only 20 amino acids, so 64 possible combinations, but only 20 amino acids plus the termination codons. That means there's a lot of redundancy in this code. In other words, multiple sequences, multiple different codons can code for the same amino acid. And you can see that over here in the chart, where if we start at the left and say we have a T, and then the second letter is, say, a C, and then the 3rd letter is, we go over here to the right, and we have an A. So TCA gives us serine, but so do all these other combinations. Okay, so TCT, TCC, TCA, TCG. This is a nice, neat one, because as long as you have TC, you know it's going to end up with SER. You know, it's a real simple one. But others aren't as simple, like Fe and Lu. You've got only 2 coding for each of these that start with a T, but Lu also has others that start with a C. So you can see there's just a lot of redundancy in this system. Why is that useful? That's useful, as we'll see in a little while, because if a mutation occurs, say, at the last letter here, but the first 2 letters are TC, but the mutation goes from an A to a C, for instance, at the end, it doesn't change the amino acid it codes for, which means the resulting protein that we make from it isn't going to be changed at all. It'll be the exact same sequence of amino acids, even though the codon was a little bit different. That said, if, on the other hand, it wasn't the last letter of TCA, but, say, the middle letter, the C, that was different, so that you had TTA instead, then you'd have a completely different amino acid, and that could change the form and function of the resulting protein. So that redundancy, it's not a perfect system for preventing errors, but it gives a little bit of leeway for that. It wasn't designed like that. This is the result of billions of years of evolution and mistakes and mutations that occurred. Some genes are broken, some, others weren't, and we end up with this system. That brings us to genes. So genes are made up of these codons, okay? So these 3-letter sequences line up in a row on the DNA, and the sequences that code for a single protein are what we call a gene, okay? So it'll start at, say, a letter T and end 500, 5,000, 5 million base pairs down with maybe a G. Doesn't matter really. But in between, you're going to have these combinations of 3 letters that you can that will translate into amino acids. Excuse me. Now, one of the interesting things about the Human Genome Project is when it was first proposed, we were assuming, since we knew things like the C. elegans nematode only had 20,000 genes, because we had already, it's a much smaller genome, we had already sequenced it. We figured we would probably end up having around 100,000, because we're a much more complex. The nematode is a very simple roundworm. Its brain has all of something like 380 or so neurons in it. It's a very, very simple, very biologically simple organism compared to humans, and mammals, and so forth. So we figured we'd probably be somewhere around 100,000. When the results came in, though, the first results looked like around 30,000. Now they've been refined down to, I think, less than 25,000 genes in the human genome. So only 5,000 genes are separating us and this incredibly simple life form. And what that means is that there's a difference in the way that the genes of the nematode work and the way that our genes work. And we'll come to that in a little bit. But the point is that there's a special trick I'm going to show you at the end, which is how our genome, how our genes can be expressed in more than one way. Okay. First, so we got to talk about what this process of protein synthesis is. How do we go from this series of codons to actually linking amino acids together into a protein? How do we bridge that gap? Especially because those codons are trapped away in the nucleus where only DNA and RNA and amino acids can be. So how do we get that information out to the ribosome to actually do this? Well, there are two main stages to this process, okay? The first is called transcription, and you can think of that simply as copying those instructions, copying that gene, making a copy that will travel out and deliver the message to the ribosomes. The second is translation, which is taking the message and turning it into an actual protein. So those are the two main stages. I want to start with transcription. There's a great animation of this on the course website, link to it next to this video. I encourage you, it's not required, but I encourage you to watch it. It's really neat. Transcription starts off a lot like replication, right? The DNA sort of uncoils and splits, but it only splits in a part. It doesn't split all the way. So just that gene kind of splits apart, right? And then an enzyme comes in that assembles a piece of RNA based on that template of the gene, okay? That RNA then gets transported out of the nucleus, and that's where translation will occur. So transcription is happening here. Here's your DNA inside this big green nucleus, and an enzyme has come along, caused this particular gene to unzip in the DNA, and another enzyme has come in and has started assembling copies of this gene using one of the sides of the DNA as a template. Those copies are made of a special kind of RNA we called mRNA, for messenger RNA. The mRNA, because it can slip in and out of the nucleus, right? It's one of those few things that can go in and out of the nucleus. It goes out of the nucleus to the site of the ribosome, and that's where translation begins, the actual assembly of the protein. So, translation has a couple of steps. First, the protein synthesis is initiated, okay? When the mRNA binds to the ribosome will read the first codon and will attract a tRNA, that is a transfer RNA, which is just a little tiny snippet of RNA that is attached to the corresponding amino acid. The snippet of RNA is like a codon. It's actually an anticodon. It's the opposite of the codon that's been read by the ribosome, so that they'll fit together, right? So the anticodon is connected to an amino acid, and it brings it to the site of the ribosome. And we can see that here, right? It's starting, the mRNA came in this way. When the green side came into the mRNA, it read that first set of 3 letters there. And then a corresponding piece of tRNA came down. So just like you can see here, this tRNA matches the codon that it's reading right at the moment. It comes down. The green thing here represents the amino acid, and it attaches it to the previous amino acid chain that has been made, and then it releases, and the next one comes down, and the process keeps going. All right. So next codon is read, same process as before, and then we keep going until the termination codon is reached. So one of those 3 termination codons, they don't code for an amino acid. They simply tell the ribosome to stop and to release the protein out into the cell to do what it's going to do, and to then send the mRNA to go out, and where other enzymes will break it back into nucleotides that can be recycled. It's a nifty little process. Here's just like another representation of it. Again, sort of giving you a better idea of like, okay, the anticodons and the codons match up. Now remember something I said about the structure of DNA. One of the main differences, there are 2 main differences between DNA and RNA. DNA is double-stranded, right, as you can see over here. But RNA is just a single strand. In addition, DNA uses the letter T as a nucleotide base, but instead of T, RNA uses U, uracil. So, bear those 2 differences in mind. They are one of the key places where students slip up in terms of the process of translating proteins. Okay, as I said before, we've got this problem of genes being expressed in more than one way. How does that happen? Well, in animals like humans and most mammals, our genes have different sections to them that may or may not be replicated during this whole process of creating the mRNA. There are sections called introns that are removed before translation takes place, and there are other portions called exons that are allowed to remain and are actually expressed. Depending on the combination of introns and exons that are either taken out or left to remain, you get different proteins. And so a single cell can produce many different proteins. This process is called alternative splicing. And it looks a little bit like this here. You've got, in the blue are the introns that are going to get removed. Where we are right now, so the DNA, this is what the DNA looks like. The mRNA is going to be formed, and then then there's going to be a protein structure called a spliceosome that's going to sort of take out the introns in between these, and then reassemble, depending on what protein is going to be built. It'll reassemble different combinations of the exons. So if you take exon 1, 2, and 3, you may get protein A, whereas if you combine exon 1, 2, and 4, you may get protein B, or you will get protein B, I should say. Again, there's a great animation of this on the course website. I highly encourage you to take a look at it. It's a little easier to visualize when you see that animation. It's a little hard to splice it into this movie right at the moment. I'm learning as I go here. So, but I hope that answers a lot of your questions. If you want to practice, or you're a little unsure about the process of protein synthesis, I've got an activity sheet that you can do. It's not graded. However, if you do it, it'll be clear whether you get it right or not. Okay. The sheet basically gives you instructions for how to decode messages that have been encoded into DNA. And by message, I mean literal messages in this case. They've taken and assigned letters to each of the amino acids, and therefore, as you go through the process, if they will give you a sequence of DNA that you will then have to transcribe into mRNA, enlist that, and then you'll have to translate that into a protein. And then when you translate it into the sequence of amino acids, you'll note the letters that are assigned to each amino acid, and those letters will spell out a message. It will be clear. It won't be nonsense. So if you end up with gobbledygook, something went wrong. If you don't end up with gobbledygook, great, you did it right. I'm happy to look at them if you're having trouble, so just let me know. We can meet in office hours or whenever. But it is just this, like I say, this is completely optional, but it is really helpful for understanding this process of protein synthesis, because it isn't always intuitive. Okay. That said, our next video is going to be the last one on DNA. And actually, we're going to be talking sort of one level up. We're going to be talking about cell division, and specifically the process of meiosis and how sex cells are formed, because it's the relation. Remember, mutations that occur in somatic cells aren't passed along, so they don't play a major role in evolution. Mutations that occur in sex cells, however, are passed on to the next generation, which means there's a possibility of the trait that a trait that has been selected for being maintained in the population in the next generation. So understanding meiosis is going to be key to understanding patterns of genetic transmission between the generations later on. It plays a role in evolution. So until then, take care of yourself, and I hope you have a great day. Bye.